Assalamu alaikum students. Today we are solving May-June 2020 paper 6 variant 1. Students, my screen is in front of you. May-June 2020. Let's start with the question number 1. A student used the apparatus shown below to investigate respiration in some small insect. The apparatus was sealed with the clip so that any changes in the volume of a gas in the apparatus cause the bubble of a colored liquid in the capillary tube to move. Here they are showing the apparatus. In this one, a substance is there that is responsible to absorb carbon dioxide on a wire gauze. Small insects are there. The one end is completely sealed with a clip. Other end has a capillary tube that has a colored liquid with the ruler. Now, suggest what happened to the bubble of a colored liquid as the insect in the test tube respire. Definitely, this bubble will move towards the insect because it is 100% clear that when these insects respire, they will use the oxygen present inside the tube, and the carbon dioxide produced will absorb by this one. So you can write here that it will move towards the insect, or you can write it moves towards the left, right? Now, using the apparatus in the diagram, the student read the starting position of a bubble of a colored liquid. She recorded the reading from the left edge of a bubble. She left the apparatus for 60 minutes, then recorded the final position of the bubble. Here they are showing you the two scales. The diagram shows the starting and final position of the bubble in a space provided. Record the reading for the position of each bubble when you are using the ruler. Then you will find out that this is 6.8. As centimeter is already mentioned, so there is no need to write down the unit. And after 60 minutes, it is 5.6. Now, next is calculate the distance the bubble moved in 60 minutes. Definitely you have to minus, and you will get the answer of 1.2 centimeter. Here they are giving you 2 marks. It means 1 mark for the calculation and 1 mark for the unit. Calculate the rate of a movement of a bubble in millimeter per minute. It means you have to multiply 1.2 into 10 to convert it into millimeters. So 12 millimeter is the conversion. 12 divided by 60, you will get 0.2 millimeter per minute. So answer is 0.2. Now, next is explain why carrying out the experiment several times, which improve the reliability of the result. Reliability means to avoid the anomalous result. It means, or you can write to avoid human error. Or secondly, it will help you to find out the mean or average. Suggest a suitable control experiment for this investigation. Control means you have to go in an opposite direction. It means they are using living insect. You have to use the same apparatus with dead or without insect, or instead of insect, maybe you can use the same apparatus by using with the help of marble, right? So there is no insect, no respiration takes place. Suggest an environmental factor outside the apparatus that might affect what happens to the bubble of a colored liquid. Definitely, if temperature increases, then it will affect on the respiration of the insect, or maybe due to high temperature, the bubble will evaporate. So temperature is this one. Describe how the student might ensure that this factor has no effect on the bubble of a colored liquid. It means how we can control this temperature. We have two ways. Either we can go for a water bath. It means place this test tube that is carrying insect uh, in a water bath, Or secondly, student has to perform this experiment in a controlled environment. The insect produce carbon dioxide as a result of respiration. Suggest and explain what would happen to the bubble of a colored liquid if the experiment was repeated in the chemical which absorb carbon dioxide removed from the apparatus. Let's move to the page number 1, where they are showing that this liquid is there. Now they are saying, okay, this liquid, they are removing this one. Now simple experiment takes place. If you check, if you remember the respiration equation, oxygen breakdown glucose produces carbon dioxide in water. If you balance this equation, you can easily find out the molecules oxygen and molecular number of carbon dioxide is same. So it means if all the insects are using 5 molecules of oxygen, they will produce 6 molecules of carbon dioxide. So it means inside the test tube, number of bubbles remains same, right? So there may be, you can say that, okay, answer will be, okay, bubble doesn't move sometimes, you can say, or it will move it will move slowly, right? And suggestion, it depend on you. Either you are writing that it is not moving at all, or you can write that, okay, small movement is there, and the reason behind this, that number of oxygen and number of carbon dioxide molecules are same. So inside the test tube, all the number of molecules remain same. Now move to the other question. The student wanted to show that the insect produced carbon dioxide. The diagram shows the apparatus shoes. When the carbon dioxide is bubbled through lime water, it turns milky. Now they are giving you the solution, giving you a hint also. 3 test tubes are connected. Suggest and explain the result you would expect after the apparatus was left in for 30 minutes. After 30 minutes, definitely this lime water will turn less milky, because less concentration of carbon dioxide present in the air. Air contains, if you remember, 0.04% of carbon dioxide is present. So definitely after 30 minutes, when air will pass through the test tube 1, it will turn less milky because some of the carbon dioxide is present there. When this air passed through this second test tube, where insects are producing carbon dioxide, when this air rushes from this to this test tube, and this C will turn more milky because there are more carbon dioxide produced by the test tube B. That is why it turns milky, right? Now students, move to question number 2. The photograph shows the cut surface of a lemon fruit. In the space below, make a large diagram of a cut surface of the lemon as it appeared in a photograph. For the 4 marks, you have to draw the diagram. When you are drawing the diagram, as we all are students, so at that time, your main concern is, first of all, when they are saying make a large diagram, so it should be large enough that anyone can easily identify that it is a large. Second one is, you have to main focus about the outline. Outline should be sharp, should be clear, it not hazy, not double, it should be neat and clean. Second one is, you have to concentrate on the proportion. Look at this one, this line, then we have this white portion, then we have this flesh portion area, then in the center we have whitish portion. So when you are drawing, you have to concentrate on all the portions. As it is segmented fruit, so you have to 5, 6, 7, 8, 9, 10, 11. You have to concentrate on its segment also. Okay. It is not 100% round. So if you want to use compass to draw the round structure, but you have to be concentrate that it shouldn't be with the compass. You have to make a freehand drawing. Okay, now move to this one. On the photograph, draw a straight line between A and B and measure and record the length of this line. Okay, on the photograph, it means examiner need that you have to measure, you have to draw a line from A till B, and it should be with the ruler. Okay, it should be with the ruler, and you have to measure. As I'm using this screen, suppose I am saying that it is 5.5 centimeter. Suppose, let me write here, suppose. Okay, they are saying you have to write in a millimeter. So 5.5 into 10, I am getting 55 millimeter. Okay, now on the actual lemon that was photographed, the measurement between A and B is 6 centimeter. Okay, use your measurement in B1, this is B1, to calculate the magnification of the lemon in the photograph. Okay, in this question, we solve our question according to examiner, it should be in a millimeter. So first of all, we have to convert 6 centimeter into millimeter. It means 16 into 10, 60 millimeter, right? Now come to the magnification formula. Keep one triangle in your mind, that I am triangle. I is always on top. So magnification formula is, you have to write down the complete formula. Magnification is equal to image size divided by actual size, right? Image size is 55 millimeter and actual size is 60. You, whatever the answer is there, you have to write down, and there is no unit for the magnification, only cross sign or time sign is there. So whatever the answer is there, you have to write down here. For this 3 marks, 1 mark is for the conversion, 1 mark is for the correct formula, and 1 mark is for the correct answer. C part is, a student tested some juice from the lemon for the presence of reducing sugar and protein. Name the testing reagent she used for this test. Reagents are required. Reducing sugar is Benedict reagent. And here for the protein, it is biuret. Students, when you were studying reagents, okay, when you were studying these chapters, this chapter where Benedict and biuret test were there, maybe there is a chance that you are getting the name of these proper chemical name for this Benedict and biuret reagent. When they are asking reagent, limited to this one, and the formula is not in the syllabus, so don't go for this one. State a suitable temperature at which each test tube should be done. Reducing sugar test, for reducing sugar test, we need water bath, and it mostly we are performing this experiment at 80 degrees Celsius. Protein test should be done easily without heating. It is room temperature, right? For the result of her test, the student concluded that the reducing sugar test was present in the lemon juice, but protein was not present. State the result she observed. Okay, if reducing sugar test present, then student may get orange, yellow, green, or brick red color. For the protein, when they are saying if protein was not present, so it means blue color remains same. Okay. The student then tested the lemon juice for the presence of fats. Explain why she ensured there were no naked flame in the laboratory when she did so. Fat test is emulsification test. Ethanol is required. Ethanol or alcohol is needed, and it is highly flammable. right? So 2 marks is what substance and why. Okay, D part is another student investigated the concentration of reducing sugar in lemon size using different method. First, he tested solution of different concentration of the reducing sugar glucose. He used an instrument called a calorimeter to measure the concentration of glucose. The lower the number on the calorimeter reading, the higher the glucose concentration. His results are shown in the table. Glucose concentration and calorimeter reading in arbitrary unit is there. On the grid, construct a line graph to show the relationship between glucose concentration and the calorimeter reading. Jot your point with the ruled straight line. I will come to this point later. First of all, we have to know it is a 4-mark question. You have to identify which factor will come on axis and which one is on y. The first column mostly is on x-axis and second is y-axis, right? You have to make a straight line for x-axis, that is, and you have to copy down the whole glucose concentration percentage. You have to write down here, and for the y-axis you have to make a straight line here, right, and write down the whole reading, calorimeter reading, arbitrary unit. Okay, you have to write down full information here. Okay, 1 mark is for the axis labeling. Both the axis should label properly. Then you have to go from 0 till 2. It means you have more space. Wait, sorry, let me move it here. Okay, from 0 till 2 you have to move here. You have to use more than 50% of your grid. Okay, 0, after 10 digits you can go for 0.5, then 0, then 1, then 1.5, then 2. Make a distance of 10 small boxes. Here you can go from 0 till 9.5. It means you can it's up to you. Either you are using, after 5 boxes you are using 0.1, then 0.2, then 0.3, so on and so forth. Keep it in your mind. Whenever you are filling the graph, you have to use more than 50% of a grid. Now, one thing, suppose your point reach here. So there are 2 ways to identify, to locate your point, either this way, or suppose it is your point, then you have to make a cross on this point. Okay, these 2 authentic points are there, ways are there to plot your graph. Then they are saying you have to use a ruled lines. It means you have to use a ruler. Okay, now you have to draw a graph yourself. The student then tested some lime juice and obtained a calorimeter reading 0.65 arbitrary unit. Use graph to find out the concentration of glucose in the lemon juice. Simplest is this one. When you are plotting here, definitely there is some point that is what they are asking, 0.65. Suppose on a graph my 0.65 is here, right? On a line you have to find out what is the concentration of glucose. This you have to show on a graph. For this one, you have to show properly on your graph and correct value from the graph, and you have to write down. Is it clear, students, everyone? Now, this is the last question for this paper. See you in the next. I will show you another paper, inshallah.